now let's talk about meiosis. Okay. So our last type of division is called meiosis, or some people may pronounce it as meiosis. What do you know about meiosis? Well, if you've been paying attention, we've mentioned it a couple times. So one thing you would note is that for meiosis, germ cells undergo, and then the end result is a gamete. This is a form of sexual reproduction, which is different than most things that we've been talking about. And then there are 4 different cells created. And the purpose is going to be for variation and reduce the chromosomes, because these cells are haploid. Now, let's look at the cycle. So, as you see, the first thing you should notice is it's not quite a cycle, like we said, for mitosis. So once these cells come out, they cannot re-enter this process again. Another thing you should see is that we're doing this twice. So we go through all the phases we talked about, and now look, they're going through phases again. So you start with 1 cell, you're ending up with 4 cells. Each of these cells has 2 copies of each chromosome. Each of these cells only have 1. Okay, so that's how we're reducing the number. And then there's variation, because if you look here, these chromosomes don't look like these chromosomes, and the chromosomes in each cell are different. Okay, so meiosis is not a cycle. You start with the germ cell, you end with the gamete, and you get half the chromosomes that you start with. You go through this thing twice. Okay, let's look at it in more detail. So I want you to notice that the phases are the same. The same phases that we talked about in mitosis are also in meiosis. Each phase does happen twice, but there's only one interphase. You only do interphase once. So you only replicate the chromosomes once, but then you go through everything else twice. Another thing I want you to notice here is that's crossing over. What is that? That's this exchange of genetic information between the 2 chromosomes. So if we go back a slide, you notice that the chromosomes were blue and orange, right? But when crossing over occurs, you have pieces of blue on the orange and orange on the blue. That's crossing over. They have exchanged genetic information. Okay? So the stages are very similar. So in interphase, similar to interphase we've discussed, you're going to grow, the cells are going to grow, the DNA is going to replicate. Now in prophase, we're going to add crossing over. But the same things happen where your nuclear membrane has to break down, your spindle fibers are formed and attached to the chromosomes, and your DNA has to condense. When you get to metaphase, this looks slightly different because they're no longer in a single file line, but a double file line. So homologous chromosomes, whenever you get one from mom and one from dad, well, that copy from mom and dad of chromosome 1 are now going to be next to each other. Okay? So now anaphase, when we pull them apart, we're going to pull apart those homologous chromosomes. And then telophase and cytokinesis are the same. So now we're going to go through meiosis 2 with 2 cells, but it looks exactly like mitosis. Single file line, you're going to pull apart these chromosomes. These chromosomes are made of sister chromatids. So here you see we had 1 chromatid, which is also equal to a chromosome. After S phase, where we replicated these chromosomes, each chromosome had 2 arms. Those arms are called chromatids. So here we're pulling apart the arms of those chromosomes. So in meiosis 1, we pull apart homologous chromosomes. Meiosis 2, we pull apart sister chromatids. And now you end up with 4 haploid cells. Haploid meaning half the number, and they're all genetically different. And then these cells can undergo something called gametogenesis, or how you make gametes. Like, whoa, what is that? So this is what the reason we actually do meiosis. So here's your meiosis, but the end result here aren't just 4 body cells. They're going to mature into sperm cells, but you have to go through this process to get those sperm cells. So you have your spermatogonium, your germ cell, is going to undergo meiosis. have this gametogenesis to become an actual sperm cell. This process begins in men at puberty. Okay? Sorry, I spelled it incorrectly. And it can essentially go until death. This is a continuous process in men. So once they start, they can just keep making them over and over again. And one cell goes into 4 cells that are very similar. Okay, so in men it's very simple, and it takes place where in men? Okay, in their testes. So in the testicles of men, they have these little cells that whose job it is to make sperm. So you have this spermatogonium. It enters the process of meiosis, where it's going to be dividing twice, meiosis 1, meiosis 2, and then maturing into a sperm. Some differences you'll notice from the spermatids to the sperm cell. It has a tail. It has mitochondria here at the base that's going to help fuel that tail, and that tail is actually called a flagella. And now in the head of the sperm cell is chromosomes and not much else. Okay, so it has to shed that cytoplasmic material and hold on to those chromosomes. And if you think back to the picture that we looked at between the egg and the sperm, you'll see that the sperm is very small compared to the egg cell. And now in women, it's a little more complicated. So you start with this oogonium. And now this is made, like a woman is born. So at birth, they have these oogoniums. So this process starts during embryonic development in women. And when does it stop? Okay, when do we signify women can no longer make babies? It's menopause. This is a discontinuous process in women because when do they release an egg? Once a month, right? So it stops at birth, and then monthly, 1 egg will be released. Now, what you're really releasing is this spermatogonium. So this process is arrested here. Okay, so we arrest this process in the primary oocyte. When it's about time for ovulation, as the hormone levels are going up and down, we're going to start the process of meiosis, and then we're going to stop again. So this secondary oocyte is actually what we consider an egg when we ovulate. This now is going to travel down from the fallopian tubes into the uterus with hopes of being fertilized. If it's fertilized, this process continues and a mature egg cell is made. If it's not fertilized, this process never continues, and you get what we call a period collectively, and that's after this has left the body, and the body is no longer need to be prepared for fertilization because it's not going to happen. So this process is discontinuous. So during embryonic development, the woman has her oogoniums. These oogoniums enter meiosis and stop during prophase. So in this primary oocyte phase. And every month now, one of these oocytes starts the process up again and then is halted at ovulation. A secondary oocyte is ovulated. If fertilization happens, it finishes the race. If it doesn't, it's expelled. Now, another thing to note here is going to be these secondary, these polar bodies here. So these smaller cells. So cytokinesis here is uneven in that what's going to become the egg cell has a lot more cytoplasm than these polar bodies. Okay? The purpose of these polar bodies is to get rid of that extra DNA because we need to reduce this. So notice here, 1 cell here, 1 germ cell equals 1 egg cell. You only get 1 egg cell. So 1 cell in men gives you 4 sperm cells. 1 cell in women gives you 1 egg cell. And then these are discarded. Okay, their purpose is to just get rid of that extra chromosome material. And so I'm confident that you can back up and compare and contrast mitosis and meiosis, and you can also compare and contrast spermatogenesis and oogenesis.